Let's look at SpongeBob and Patrick. Each of these individuals are diploid, meaning that they have two of each particular chromosome, one from each of their parents. First, let's look at their DNA before meiosis occurs. Their DNA is not condensed and looks like a huge mess. Let's look at SpongeBob first. Here are SpongeBob's chromosomes just after S phase, where each of the homologous chromosomes duplicated in order to have two sister chromatids attached to each centromere. The DNA condensed into chromosomes starting at prophase 1 of meiosis. These homologous chromosomes might not look identical now, but just flip the one on the right by 180 degrees. It is important to get each analogous portion of each homologous chromosome to pair together in order for synapsis to occur successfully. During prophase 1 of meiosis, synapsis occurs and the non-sister chromatids of the two chromosomes can cross over to exchange their genetic material. We will begin crossing over between the J and K regions of the chromosomes. I will start by pulling the left sister chromatid from the left chromosome, and next I will work on separating the next sister chromatid of this chromosome. However, in this case, when we get to the J region, we must follow the crossover to the other non-sister chromatid, following the path that I am indicating with my finger. Now, I will pull the right sister chromatid from the right chromosome. And next, I will work on separating the next sister chromatid of this chromosome. Remember that in this case, we must follow the crossover as indicated by my finger and the X-like rope figure. Now let's look at Patrick. After DNA condensation, the homologous chromosomes can be seen MLI centromere JKHG and GHI centromere JKLM. Contrary to SpongeBob's homologous chromosomes, Patrick's are not the same, even if we did flip them by 180 degrees. In Patrick's homologous chromosomes on the right, we can see that a pericentric inversion has taken place relative to Patrick's left chromosome. Since this is the case, it would be impossible to pair the homologs together without creating an inversion loop to allow similar portions of the homologous chromosomes to pair during prophase 1 of meiosis. Let's draw a pericentric inversion loop to pair them together. Now let's indicate where on the inversion loop the crossover would occur between the J and K regions. Just start and follow the lines around and pair the regions of the chromosomes together that are on the same line. Let's start at the very left on the topmost line. Take M, L, I, a centromere, J, K, H, and then G. So that is our first possible gamete. Now let's start on the next line down on the left. We have M, L, I, a centromere, J, and then we will need to cross over to the next innermost loop and follow that path. Continuing, we will have K, L, and then M. Now we will start at the third line down. We have M, L, K, J, a centromere, I, H, and then G. Now we will need to start on the second row down on the right side instead of the left, since we have already traced all the lines that start on the left. We have G, H, K, and then we must follow the crossover as indicated by my finger to the next innermost loop. Continuing, we have J, a centromere, I, H, and lastly G. Gamete 2 from Patrick has a duplicated M and L and a deleted G and H, and Gamete 4 has a deleted M and L and a duplicated H and G. Only 2 of Patrick's gametes would produce viable offspring, and all of SpongeBob's would. SpongeBob therefore has a greater fitness than Patrick.